We are immensely pleased to have Honorable Vice Chancellor, Sir Lieutenant General V.K. Sharma, recipient of Ati Visis Seva Medal, respected Pro Vice Chancellor, Sir Professor Dr. M.P. Kaushik, Deputy Pro Vice Chancellor, Sir, revered dignitaries, Registrar, AUMP, esteemed deans, directors, HOI, HODs, faculty members, staff member, and our valued delegates of this event. And my dear students, I, Dr. Jovita Kanodia, welcome you all. A very warm welcome to our National E-Conference on Novel Drug Delivery Strategy for the Treatment of Cancer, organized by Amity Institute of Pharmacy, Amity University, Madhya Pradesh. Now, I would like to welcome our chief guest or guest speaker virtually present with us. I extend my warm welcome to the Director, Amity University, Madhya Pradesh, Dr. S. Mohana Lakshmi, to this event. Now, I would like to call the organizing secretary of the conference, Dr. Neeraj Mishra, for his inaugural speech. Sir, please.
Yeah, good morning, all of you. Yeah, myself, Dr. Neeraj Mishra. Today, I'm here to give a welcome speech on the conference theme that is based upon novel drug delivery strategies for the treatment of the cancer. Cancer has emerged a leading cause of death worldwide. Although conventional chemotherapy has been a keystone to combat cancer, but it is also associated with the normal cellular toxicity. Due to lack of specificity, conventional cancer treatment often cause severe side effects and toxicity also. So current challenge of anti-cancer drug development includes site-specific delivery with low systemic toxicity. Drug development includes site-specific delivery with low systemic toxicity. The development of multiple drug resistance (MDR) is one of the major challenges. Multiple drug resistance (MDR) is one of the traditional chemotherapy. And the multi-drug resistance, because of it also developed because of the superbugs. Superbugs, yeah. Superbugs, they're also called antibiotic resistant bacteria. In the world, approximately 70 lakhs of people die every year due to the problem of the MDR. Recently in Britain, a scientist had discovered synthetic antibiotic known taxibactin analog, which is very selective in the killing of antibiotic resistant infectious cells, superbugs, without damaging the normal cell. And it could be help in the fighting against the coronavirus by the using of the drug repurposing. Novel drug delivery options like liposomes, dendrimer, nanofiber, quantum dots, CNT, and metal nanoparticle emerge a promising approach, and thus are expected to provide a newer and advanced avenue in the cancer treatment. The conference will also provide a platform to exchange the latest innovative ideas and research findings of the budding researchers in the area of the cancer treatment. I wish all of you a great enlightenment in the National E-Conference. And on the note that let us commence the day by the seeking of the blessing of Ma Saraswati, the goddess of knowledge. And I request all the dignitaries to kindly join us for the auspicious ceremony. Okay, thank you. Thank you, all the esteemed dignitaries. Now I would like to call Dr. Jovita Kanojia for to give a brief introduction of our Honorable Vice Chancellor, sir. Okay. Dr. Jovita, please.
Thank you, sir. I would like to welcome our chairman of this conference, Lieutenant General V.K. Sharma, AVSM, with huge round of applause. Thank you, everyone. It's my honor to introduce the Honorable Vice Chancellor of Amity University, Madhya Pradesh, Lieutenant General V.K. Sharma, AVSM. With his vast experience of 4 decades of distinguished service in Indian Army, he brings to the university progressive academic leadership and rich administration experiences. Lieutenant General V.K. Sharma holds a postgraduate in engineering from Pune University and master's degree in management from Osmania University, Hyderabad. He was a part of Operation Rakshak and Operation Vijay during the Kargil War, which raised the dignity of our country in front of National Front. For his dedicated and commendable service to the nation, he has been confirmed with the Ati Visis Seva Medal from the Honorable President of India under his leadership. Amity University, Madhya Pradesh promotes excellence in higher education for a vibrant and inclusive society through a knowledge creation and dissemination. Sir, I would like to invite you for your opening remarks. Please, sir.
Thank you very much, Jovita. So, Dr. Anup Kumar, Dr. Sharad Kumar Pandey, Dr. S. Mohana Lakshmi, the Director of AIP, Dr. Neeraj Mishra, Dr. M.P. Kaushik, Mr. Rajesh Jain, all the faculty members, participants, and students who have joined in physical mode as well as in online mode. This conference is being conducted in a hybrid mode, and the topic which has been selected is so apt because cancer, although it is curable, but it is curable only if detected early. The main issue of cancer treatment lies at what stage it is detected. If it is detected while it is in stage 1 and stage 2, the chances of cure are about 80 to 100%. But if it is detected when it is in stage 3 or 4, the chances of survival are much reduced. In spite of the best type of treatment and drug delivery systems which are available today. Now, what exactly is cancer? Cancer, in a layman's term, is unprecedented growth of cells at a rate which is much higher than the normal growth. It results in tumors which are painful, which block the passage of flow and liquids in the body, which cause impediments, and the growth is almost unlimited. It keeps on growing till such time it causes death. And for cancer, now it has been realized nothing better than prevention. Prevention is the best cure for cancer. And to all students and all participants who are listening to me, I would like to talk more about prevention of cancer. Now, cancer can be prevented if you are doing regular exercise, if you are providing enough oxygen to your system, because the food that we consume contains primarily hydrocarbons and proteins, and these to be converted into energy, which is called the metabolism, it requires oxygen. And the oxygen gets supplied through the lungs. But the requirement of oxygen generally falls short, resulting in excessive fat accumulation in the body, and excessive sugars, excessive fat, excessive proteins, all these things in the body will lead to cancer. So therefore, the aim is to burn down the excessive sugars, the excessive fat, and to regulate the proteins and the movement inside our body of the food that we consume, avoid sugars or keep them to the limit, avoid salt as much as possible, do regular exercise, resulting in more intake of oxygen, and if one is following a healthy lifestyle, the chances of getting cancer get reduced. Now, the cancer, in some cases, it is also hereditary in the sense that the cells or the genes, they have information stored in them about the disease also, which gets activated at appropriate time when they become active. So at that time, the cancer cells start multiplying, resulting in the tumors. The tumors have been successfully cured earlier since time immemorial in India through surgeries which have been performed. The details of those surgeries are also available in our scriptures. But the first surgery, which is supposed to have been performed without anesthesia, was in 1809, if Google is to be believed. And that was done by Mr. McDowell. He removed it with a tumor, ovarian tumor, without the anesthesia. You can imagine how painful it would have been and how much time to recovery would have taken place. But then after anesthesia was discovered, exactly after 37 years in 1846, the operation started being done of removal of tumors by using surgical instruments under anesthesia, which was much more humane process. And then, of course, Röntgen discovered X-rays in 1895, and after that, the radiation treatment of cancers became prevalent. But in radiation, the problem is that it is not only the cancer cells which get eliminated or killed, but the useful cells, the normal cells which are required for functioning of the human body also get killed. And therefore, it results in lowering of vital components and lowering of metabolism and functioning of the body. The digestion system, the respiratory, everything becomes weakened because the normal cells are also getting killed along with the cancerous cells. That is the biggest drawback of radiation. And same thing, after the chemotherapy has been discovered in the beginning of the 20th century, same thing again with chemotherapy is very heavy dose of antibiotics which are given. And antibiotics, what they do, they create antibodies inside which try and fight the cancer cells in a more effective manner. And of course, the chemotherapy also results in the diminishing of normal cells. So therefore, the side effects are tremendous. It results, anybody who is under chemotherapy undergoes a loss of hair, a loss of appetite. The functions like kidneys and liver, they get affected because of the useful cells getting killed along with cancer cells. And that is how the things stand today. So it's in fact, you can say, four-legged system, that is surgery, radiation, chemotherapy, and followed by other medicines, heavy antibiotics, which are also given. Tobacco is a main factor for cancer, which is known to all of us, but yet some of us continue to consume tobacco in unlimited proportions, knowing fully well that it is going to result in cancer of the mouth, throat, etc. Drug delivery systems is what is the necessity of today, and now the drug delivery systems are being used which can specifically deliver the drug using nanoparticles, which are still at experimental stage, not fully authenticated, and the procedures is not that prevalent and not that much that it can be followed in routine at most of the hospitals. And in that, the nanoparticles are so small in size that they can interact and they can carry the medicine to the exact spot, to the cancerous cells, so that the cancerous cells can be killed and eliminated. But the nanoparticle itself remains in the bloodstream or in the body system after it has done its job. They mix with the bloodstream, they mix with our normal cells, and how they are going to behave over a long period of time is still not known, because the side effects of this type of systems are not yet known, and much more study needs to be done. In today's National E-Conference, we have some of the best papers which have been received. We have experts who will be talking on the subject, and we have persons who will be presenting their research work in parallel sessions. And I think it's going to be very useful for students of biotechnology, for students of pharmacy, and for students of nanotechnology, and those who are dealing with medicine, to see which are the novel methods which can be used for delivery strategy for the treatment of cancer, which is the topic for today. So I think the topic is very apt, and we need to do this and understand these systems for the betterment of humanity. The whole aim here is to ensure that the humanity is made disease-free, or the disease which comes can be eliminated in a short duration of time effectively by giving those drugs which have least side effects and which can cause the elimination of cancerous cells and the cancerous portion and stop further cancerous growth, even though it's malignant, in a time-bound and an effective manner. Also, cost is another criteria and consideration for a country like India. Getting very high-tech technology for drug delivery systems, which is cost prohibitive, which is out of reach of normal human beings, is of not much use in India. We must remember, all scientists, those who are working on this, that we require cheaper systems. Our people here just cannot afford very luxurious or very expensive treatment, but the treatment is required by all. Unfortunately, I don't think there's any vaccine which can be taken and you can be sure for the rest of the life that you will not get cancerous cells or you will not get cancer. And the cancerous forms differ, like the blood cancer, the leukemia, what you call, that is different. In ladies, the breast cancer is prevalent to quite an extent, which gets detected. Sometimes if it is late in detection, then it causes problems. And other type of cancer, cancer of the mouth, cancer of the pancreas, cancer of the liver, so many types of cancers which are there. So different types of systems are required, drug delivery for each different type. And personalized drug delivery system is another thing which is in today, because each individual is an individual by itself. Our characteristics are different, our fingerprints are different, and each individual would require a different type of treatment rather than having one standard treatment for everybody. And that is what is called, comes under the personalized treatment. But as I said, these are likely to be very expensive. Hopefully, with time, the cost will come down. But the researchers, while doing any type of research, must keep this factor in mind about the cost for the product that they're going to suggest. So I wish this conference all the very best, and I'm sure the students and the participants are going to benefit from the vast knowledge of the speakers who are lined up today and tomorrow also. And I wish, and the organizers have worked very hard to coordinate various aspects of the conference, to get the papers, to do all sort of administrative work before the conference could actually happen. So my compliments to Dr. S. Lakshmi, the director, and her team who have worked relentlessly to make this conference a great success. I wish this conference all the very best. Thank you very much, and Jai Hind. Also, I'd like to mention that I would like to leave this conference sooner. Much I would have desired to attend, but because of another commitment, I think I will log out maybe after another 5 minutes or 10 minutes. But I wish all the very best to all of you. Thank you very much, Jai Hind.
Thank you, sir, for the word of wisdom and for setting a perfect stage for today's deliberations. Now I would like to call Dr. M. Pratap for a brief introduction of today's guest speaker, Dr. Anup Kumar. Sir, please.
So good morning to everyone. We are delighted to have a gracious presence of our guest speaker at today's conference, Dr. Anup Kumar, faculty in the Department of Pharmacology and Clinical Research, Delhi Pharmaceutical Research University, New Delhi. He has completed his master's from Jamia Ahmedabad University, Delhi, and PhD from Birla Institute of Research Technology, Mishra, Ranchi, as DST, Inspire JRF, and SRF Fellow. Earlier, he worked as an assistant professor in the Department of Pharmacology and Toxicology, National Institute of Pharmaceutical Education Research, NIPER, Raebareli. He also worked as officiating head and associate professor in the Department of Pharmacology, ISF College of Pharmacy, Moga, Punjab. He also worked as a research scientist in the Medical Affairs and Clinical Research in the Department of Sun Pharmaceutical Limited, Gurugram, and Translational Health Science Institute, Faridabad, India. Currently, he is an expert member of the Board of Studies of the Clinical Research Program of IKJPT University, Jalandhar, Punjab. And he is an expert member of Drug Discovery Hackathon 2020, initiated by AICTE and MHRD, Government of India. He has authored more than 70 research and review articles, and he published 10 book chapters in the International Journal of Repute. He is also an author of 2 books. He guided 42 postgraduate research students and 1 PhD students. He is also filed 2 patents. He also worked as a member secretary of the Institution Animal Ethics Committee and member of Human Ethics Committees and coordinator of the ADR Monitoring Center. So with this brief introduction, now I request Dr. Anup Kumar to address the gathering. So please take over the session. Sir, Dr. Anup Kumar, kindly take over the session, sir.
Thank you so much, sir, for your kind introductions. So, am I audible?
Yes, sir. You're audible.
Okay, fine. So, first of all, I would like to thank you for kind introductions. So, meantime, I'm just opening my presentation. Please confirm when slides are visible.
Is it visible, sir?
Okay, thank you so much for confirming.
Sir, kindly make it as a full screen, sir. Share screen.
Yes, I am doing.
It is
Okay.
okay.
Is it okay now?
It is okay, sir. Thank you.
Okay, so thank you so much and good morning, everyone. And first of all, I would like to thank Chancellor Sir, Vice Chancellor Sir, Neeraj Misra Sir, and all the management committee of MIT University Gwalior for giving me this opportunity to be a part of this wonderful conference. So for next 45-50 minutes, I am going to talk specifically on pharmacoeconomic studies of novel drug delivery systems in the treatment of cancer. So contents of my presentation is I am going to give you the brief idea about novel drug delivery systems and what actually the pharmacoeconomic study is and why these are important with respect to novel drug delivery systems and particularly in the treatment of cancer. What are the different types of these studies and how students can perform these studies in their settings or in their institutes, and what models currently we are using to do these studies and what software, commercial as well as freely available software, and followed by conclusions. So NDDS, as it's a theme of this conference, novel drug delivery systems in the treatment of cancer. So I think in audience, everyone have heard these terms: liposome, nanoparticle, microspheres, neosome, and so on. So these are all are the novel drug delivery systems, and these have plays a significant role in almost in every disease, but particularly when we talk about cancers, these novel drug delivery system plays a significant role. And you can see some of the examples, the drugs which are in market for the treatment of cancers in as a novel drug delivery system. So you can see in the table doxorubicin in a liposomal form. There is a cytarabines, there is a donirubicins, and there might be other drugs. There are so many drugs are under clinical trials also. So why these drug delivery system is important, I think everyone knows, but I am just going to brief because these delivery system, as Chancellor Sir has also highlighted the importance of these systems, because in cancer, the treatment which we have are associated with various ADRs, and ADRs are not minor. These are major, as well as we have seen many patients with serious ADRs due to the drugs which we are using in the treatment of cancer. So of course, we need the delivery of these drugs because the delivery of these drugs are not very specific. To make it very specific at the target site, we are reducing the dose of these drugs, as well as we are increasing its efficacy and reducing the dose. Ultimately, it will increase the safety profile of the drugs. So these novel drug delivery systems, every systems have their own advantages and limitations depending upon the nature of molecules we are choosing the system, whether this medicine we can convert into liposome or nanoparticles or other novel drug delivery system. But I am going to specifically talk on another aspect of novel drug delivery systems and in treatment of cancer. So you know, in cancer treatment, the cost of the therapy is very high and it's very difficult. As sir has told, the problem in cancer is late detection of disease. The patient is already in the late stage, and in the later stage you have to give the medicine through a IV, and you have to give the medicines which have a various side effect, as well as these medicines are very costly. There are many examples where treatment cost is more than 2 crore, 4 crore, 5 crore also, and normal public or normal families can afford it. No, it's not affordable. So the development, these whatever novel drug delivery systems has been prepared, they have played a significant role. So I have just added one example to better understand about this concept, what actually the economic analysis is. So I have just added example where you can see in this slide doxorubicin. Doxorubicin is an anti-cancer drugs, and its liposomal intravenous dispersion is available, and its vial is around 47,000 rupees. And doxorubicin simple intravenous solution, not a novel drug delivery systems, its cost is around 3,000. So if I ask you the questions in the audience, which one is economical? So of course you will say, students will say, sir, the doxorubicin intravenous solution that is of 3,000 is more economical than 47,000. and 40,000, 47,000 is the liposomal intravenous dispersion. So what is it right, or we have to be analyze it in a proper way, then only we can get to know which one is more economical. By looking only on the cost data, you can't say this particular formulation is economical and this is not economical. So how to do this, how to get these answers, you have to do a pharmacoeconomic studies. So that's why pharmacoeconomic studies have a very important role when we are talking about the cancers or the disease where cost of therapy is so high. So by looking the cost of these two different formulations, you can say yes, this 3,000 doxorubicin intravenous injection is more economical. So why I am not saying this? The reason is when we are going to do the evaluations, we are not going to do the evaluation of only the cost, but we are comparing the cost in terms of efficacy and in terms of its safety. So both analyses are important. If suppose this doxorubicin liposomal intravenous dispersion, when you are using in a patient, the cost of therapy is only for a 3 months, whereas you have to use this doxorubicin intravenous solution for 1 year. So automatically, when you will calculate all these, you will do all these calculations, then you will get to know, oh, this doxorubicin liposomal intravenous dispersion is more economical to the patients as compared to doxorubicin intravenous solutions. So these concepts I am going to talk, these specifically the potential for, because these novel drug delivery systems can reduce the adverse event, which will play a significant role in new capsulated therapeutics, which result in fewer medicinal procedure, leading to reduction in personal cost. And even though when patients are using a novel drug delivery system, there is chances the patients will return to professional life at earlier, right? Whereas when you are using a conventional drug delivery systems of that particular drug, maybe it will take time to return the patients to the professional life, as well as quality of, when we are talking about a quality of life. So when we are using the medicines in cancers, as already I said, these are associated with various adverse drug reactions. The quality of life of the patients will alter. So all these factors we have to be considered, and then only we could say yes, this novel drug delivery system is more economical as compared to conventional drug delivery system. That is the one way. Another things also, you can compare between the other drug delivery systems. Suppose you want to compare liposomal with nanoparticles with other novel drug delivery systems, you can compare, and then you can come up with a answer. Yes, among these, this one is more economical or affordable to the patients. So what actually the pharmacoeconomic is, I hope many of you knows already. So it's a not only, sometimes students think about pharmacoeconomic means only we have to be do the cost calculation. It's not only the cost calculations, but you have to be compare the cost and its consequences. And consequences, you have to consider both factor, efficacy as well as safety. It's not only if suppose you are saying this medicines is of cost of 100 rupees, and same medicine in different brand is available at 10 rupees. So 100 rupees medicine is better than 10 rupees. Is it like that? 100 rupees medicine is more efficacious, more safer. Is it like that? So you have to do all these analysis to get the answer of these critical questions, and then you will decide which therapy is better for patients in terms of safety, in terms of efficacy, but also in terms of cost also. So I just yesterday put in a PubMed. You all can do it in the PubMed search item. Just I have added the pharmacoeconomic, and I got this trend in the pharmacoeconomics. So you can see year by year, pharmacoeconomic studies, number of pharmacoeconomic studies are increasing significantly. So this shows that how popular these pharmacoeconomic studies are, and they have a important role not only in the research, but also these studies will help the policymakers to make a better better decisions regarding health policies. It helps the physician to decide which medicines, which particular medicines I have to be used in this particular type of patients. So why needed in cancer treatment? As I told you, I have put it some examples, hypothetical examples. Suppose my first question is, suppose the patient is in the last stage of the cancer, and doctor has said this vial is needed, and this vial cost is around 2 lakhs or 4 lakhs. But doctor is not assure you whether by using this injection vial the patient's survival will increase for further 1 year or 2 years. That is not a surety. Doctor will say you can use it. We can't guarantee whether patient is going to survive for a 1 days or 2 days or 3 days. So suppose the patient's family member have purchased that vial and patient is survived only for 1 days or 2 days. So ultimately the patient is died, and the financial burden will increase on the family persons. So the question is, as Indians, be all are emotional. If any
Excuse me, sir. Sir, Anup,
in
sir.
our
Sir, excuse me, sir.
family is
Sir, Anup, sir.
suffering
Your voice is breaking, sir.
from
Hello, sir.
whatever it's last stage or any, right?
Anup Kumar,
Yes,
sir.
sir.
Am I audible to you?
Yes, sir. Is it now clear? Yes, yes.
Sir,
Yes, sir.
breaking. Now it is okay.
You are audible to me.
Now your voice is
Okay.
breaking.
Okay, okay. I'm checking just. Now is it okay, sir?
Now is it okay, sir? Very fine.
Okay, okay.
Thank you.
I think now it's audible. Okay. Is it audible now? Okay. So what I am telling is about this important question regard why these pharmacoeconomic studies required. Suppose any patients of cancer is in the last stage.
So please share your screen.
Okay. Okay. I am very sorry for it.
No issue, no issue.
Is it okay now?
Yes, sir. Now it is visible.
Okay. So student, what I am telling is, suppose कोई भी कैंसर का पेशेंट्स है, लास्ट स्टेज में है, डॉक्टर अगर बोलता है कि ये एक वाइल अरेंज कर दो या 4 दिन के लिए वाइल अरेंज कर दो, एंड इट्स कॉस्ट ऑफ थेरेपी इज 2 lakhs. So as I told, being a Indians, means we all are emotional, and every family member will try to arrange it and will arrange. Whether they are taking loan or whether कई बार लोग means they are अपनी तो question is कि अगर has been died. Question is, are you costly treatment to the last stage cancer patients? It's a question mark. You by are telling to the family member, they are saying ये आप अगर बाइल अरेंज कर लेते हो या ये क्लिनिकल ट्रायल में ड्रग में भी इट विल बी इफेक्टिव और मींस थोड़ा सा बहुत सर्वाइवल बढ़ जाएगा पेशेंट्स का या नहीं बढ़ेगा. So who will answer these critical questions? These are very critical questions. So these studies pharmacoeconomics will help help to answer these questions. Another
Sir, Anup sir.
I think sir is reconnecting.
very,
Yes, sir.
very sorry. I don't know why it's happening today. Okay. Very sorry. Is it okay now?
Now visible, sir.
Okay. So I don't know where I left. So I think on this slide I was there. So I am just talking about why pharmacoeconomic studies are needed, particularly in the treatment of cancer. And as everyone knows, there are various drugs are available in the market in the form of generic, as well as different brands are available. And there are confusion among public, among physicians, among pharmacists, which brand is better. So sometimes we are thinking that more cost means more effectiveness of medicines, but it's not like that. Sometimes, particularly as we are saying, generic and branded drugs have a similar bioequivalence, have a similar AUC, have a similar Cmax, Tmax. So how these are the most costly medicine is more effective as compared to lower cost medicines. So these questions will be answered by doing a pharmacoeconomic studies, which brand is better. And particularly if you want to see which novel drug delivery system is better in terms of efficacy, in terms of safety, you have to do pharmacoeconomic study. Then only you will get to know which novel drug delivery system is affordable to the patients and also efficacious and also safety, also safe. So just try to understand what is the basic pharmacoeconomic equations which we are applying for any disease. One part is cost and another is outcome. And outcome we are measuring in a three ways. One is efficacy, safety, as well as quality of life. And when we are talking about a cancer of patients, then quality of life is very important, because we know if the patient in an advanced stage of the cancer, so it's the chances of survival is very less. What we want, the patient will survive, but survive with quality of life. So that's very important outcome. So we have a two things. One is cost and another one is outcome. And we have to be compare, because comparison you have to do between the two drugs, between the two drug delivery system. If you want to compare novel drug delivery system with the conventional drug delivery systems, then only you can say, yes, this liposomal formulations of this particular drug is more economical than conventional drug delivery system. So that means maybe their cost is high. When you see the cost, you are seeing this injection cost is 5,000 rupees, whereas conventional drug cost is 2,000 rupees. What when you will do the analysis in terms of safety, efficacy, as well as quality of life, then you will get to know, no, liposomal novel drug delivery system is more economical than conventional drug delivery systems, right? So this is basic pharmacoeconomic equations, and I have just added a few cases for better understanding to the audience. This is the adjuvant therapy of HER2 negative localized disease. And when we are talking about economic analysis, we are talking about NLEM, that is the National List of Essential Medicines. So you can see in this graph, these are the medicines which we are using: cyclophosphamide, erythromycin plus 5-fluorouracil. And these are the medicines which are listed in the NLEM. These are listed in the National Essential List of Medicines. But when you replace one of the drug with other, which is not mentioned in the Essential List of Medicines, like erythromycin, erythromycins and epirubicin have a similar mechanism. They are inhibiting the topoisomerase 2. So when you are replacing that drug with this drug, you can see in this graph how much variations in the cost is happen. So when you are picking the drugs which are mentioned in the Essential List of Medicine, the cost of therapy is around 13,000. And when you are replacing one of the medicines with which is not listed in a essential list of medicines, then you can see the cost is increased up to 46,821. It's a 6 times higher. And mechanism of actions and efficacy of these both drugs are almost similar. So the question is, if physician is picking this one instead of this one, so how much financial burden will increase on the patients, on the particular family of that particular case, cancer patients. So that's one of the very simple examples which shows the importance of pharmacoeconomic studies with respect to cancer treatment. It's very important to do these analysis. This is another examples, but it's example of hypertension, so I am not going to discuss it. Types of cost in pharmacoeconomics, it's not only the cost which is involved directly to the patients, but it's a indirect cost also we are considering. We are also considering the intangible cost. So indirect cost, what cost of the drug, what is the cost of administration, consultation fees, standard care, hospital, ICU. So these are all the examples of direct cost. Indirect cost means how much earning has been lost by the patient. Patient is admitted in the hospital for 1 month. So ultimately the patient is doing work somewhere. So it's a loss of earning, cost of travel to the hospitals. So all these costs are under indirect cost. And when we are doing a analysis, these kind of analysis, we are considering direct as well as indirect cost. Yes, intangible cost also is there, but it's not possible to measure in a monetary terms. So we are not taking it, but of course there is a type of cost is intangible, like worry of families for particular patients. These all are intangible costs. So these are the different type of cost which we are considering when we are doing such kind of analysis. So there are different types of pharmacoeconomic studies. Most commonly common types: cost minimizations analysis, cost benefit, cost effectiveness, cost utility. And you can see this quadrant where you can see in the left side, cost is more, effectiveness is less. Cost is increase, but effectiveness is less. Cost is decrease, effectiveness is less. Cost is decrease, effectiveness is more. So we will discuss it in much more detail. So first, one by one, I am going to discuss about these pharmacoeconomic studies. So first one is cost minimization studies. Cost minimization studies, main important assumption is when you are considering the outcomes of two delivery system is almost equal. So in that case, you can go for a cost minimization. If there is a difference in the outcomes, so you can't go for a cost minimization. So you can see clearly in this particular slides comparison. I think everyone understood in pharmacoeconomic studies, we have to be compare two things. So in this case, we are comparing the drug delivery, one drug delivery systems with the another drug delivery systems, or novel drug delivery systems with the conventional therapy. So in that case, first type is cost minimization analysis. Cost minimization analysis we are doing when we are assuming the cost outcomes of both the treatments are equal. We are assuming these are equally efficacious, these are equally safe. So in that case, we will go with the cost minimization studies, and particularly these studies we are doing with respect to generic products, because in generic products, what is our assumption? In generic products, we have to show only, the regulatory authority is asking you just to show the bioavailability or bioequivalence of your product. There is no need to do safety studies. There is no need to perform the efficacy studies. If your formulation is same, your API is same, your method might be a little bit different. So in that case, you have to be so only the bioequivalence studies, and in that case you have to go with the cost minimization analysis, because you are assuming these outcomes are same. Another types of evaluations which we are doing is when you are considering outcomes are not equal. So in that case, you can pick up cost benefit analysis. Only difference between cost benefit analysis, your outcome you have to measure in terms of monetary benefits. Whereas in cost effectiveness analysis and cost utility analysis, your outcome will be in physical units like morbidity, mortality. In cost utility, we are doing outcome we are measuring in terms of quality adjusted life years, morbidity plus morbidity, or quality of life. So these are the different types of economic evaluations which we are doing. So one by one, I am going to discuss how you can use in cancer or novel drug delivery systems. So in cost minimization, as I told, where you are assuming the outcomes of two treatments are equal. So branded drugs, particularly we are comparing branded with generic drugs or generic drugs. So in that case, these studies are very helpful to get to know which how much cost percentage of cost variation happens between branded generic or generic medicine. So my purpose is to tell you how you can do these analysis in your institute or in your laptop. So particularly in cost minimizations, very simple analysis, you have to be calculate the percentage of cost variation. So how to calculate, how to perform these analysis in these studies, what is required? You need the cost of that particular medicine. You need the data of cost data, right? So sometime what students are doing, they are picking cost data from one mg app या कहीं और से उन्होंने cost का data लिया. That is not a authenticate sources. So from these sources you can't go for a publications. So you have to be take the cost data from the authenticate sources. So from where you will take, if you want to perform a cost minimization study, small studies, so how to perform it? You have to be take the generic medicine cost, and generic medicine cost you can take from Jan Aushadhi websites, where you can get the cost of generic medicines with price, with unit, with the particular strip. All informations regarding the cost of generic medicines you will get from these websites. That is the one thing. Another thing which is required to do the cost minimization analysis is cost of branded drugs. You know, means there are many brands, same drug, paracetamol is available in different brand, Cipla brand, Sun Pharma brand, and so on, companies brand. So another thing which is required to do is analysis is cost of that branded drug. So from where you will get the cost of branded drugs? You will get from CIMS. This is the current index of medical store, which you can refer, where you will get the cost of different branded drugs, right? And then you can use the formula, very simple formula, everyone can use it. Percentage of cost variation is equal to cost of most expensive drug minus cost of least expensive drug divided by cost of least expensive drug multiplied by 100. So you will get the percentage of cost variations. And this is a recent in 2021 we have done this simple analysis and published in one of the journal, where we have done the cost minimization analysis used in treatment of asthma or COPD. So by similar way, it can be done for any disease. In cancer also, you can pick up any delivery systems, any drugs, and perform it, and you can publish it. So these are the results of these papers, where we have compared the medicines, generic medicines with different branded, and then we have applied the formula, and we will get to know how much cost, percentage of cost variations between these branded and generic drugs. What challenges in cost minimization is, of course, the cost data. Sometimes price are updated. Sometimes there is a discount on the medicines. So these are some of the challenges. You need recent updated data of the medicines, not the older data. So all these data, these websites are regularly updated. So you can get generic drugs cost from the Jan Aushadhi websites, and CIMS is also updated time to time, current index of medical sciences. If you are not getting it, you can get in a medical store. These peoples are using these. They have a booklets. In that one you will get recent version, you can use it. That is the one part, cost minimization studies. Next is very important with respect to cancer is cost effectiveness. So cost effectiveness we are doing when we are considering outcomes are not equal, right? So we are considering two treatments are, we don't know, they are differ in their outcomes in terms of safety and efficacy. Now we want to do, if I am putting 100 rupees or 200 rupees in this particular medicines, and same medicine is also available at 10 rupees, so which one I will prefer, 100 rupees, 200 rupees, or 10 rupees, or 15 rupees? So in that case, these studies will help you to decide whether what will be the outcome, whether this outcome is better than that or not. So cost effectiveness, as I told, we have to be three things are required: cost, benefit, and safety. And cost effectiveness analysis is an analytical tool in which the cost and harms and benefits of intervention A, and you need at least one comparator to do these kind of analysis. And in cost effectiveness analysis, we are calculating ICER, that is the incremental cost effectiveness ratio. So very simple formulas we are using to do the cost effectiveness, these incremental cost effectiveness ratio. You have to present it as a ratio of incremental cost of intervention A minus cost of intervention B, and then incremental effectiveness of intervention A minus effectiveness of intervention B, and you will divide it and you will get a ICER, incremental cost effectiveness ratio. So this is a formula. You need two medicine cost A and B and divided by effectiveness of intervention A minus effectiveness of intervention B. And if the incremental cost is smaller or incremental effect is large, then ICR would be small. So you can see this from these formulas. So therefore, a smaller ICR, smaller value you are getting means it's a better cost effectiveness. So you have to be calculate these incremental cost effectiveness ratios. And after that, what you have to do after calculation of this ICR, you have to be compare it with the cost effectiveness threshold, and from where you will get the threshold value. The threshold value you will get from it vary from country to country. That is we call willingness to pay. Willing to pay is a cost effectiveness threshold of payment for a treatment, and every country WTP have a different value, and it depends upon the GDP of that country. So I have just put it the example of UK, where is a ICR criteria of this much British pounds per quality adjusted life year. So cost effectiveness of treatment A is good if its ICR over treatment B is less than this. So of course it will be a cost effective, but if it cross, then it's not a cost effective for that particular country. So cost effectiveness threshold based on the multiples of the country per capita gross, that is the gross domestic product, GDP. So this is the decision criteria in cost effective analysis or cost utility analysis. As I told you, these are the quadrant 1, quadrant 2, quadrant 3, and quadrant 4. And particularly in quadrant 3 and 4, there is a very difficult to make a decision. So in that quadrants, you have to do these analysis. If suppose medicine is costly and less effective, so easily you will discard it. You will say, no, I am putting the cost and it's not effective, so you can exclude it. And if the medicine is effective but very costly, you have to be do these analysis to make a better clinical decisions. So how to do it? That's very important. My purpose of presentation is so everyone in the audience can do these analysis whenever they get a time. They can think about and do these analysis in their project work or small kind of project in B form or in M form. You can do it and you can go for a publication. So how to do it? How to, if someone planning to do a cost effective analysis, so how to do it? So to do these kind of study is what is required. Two things are required. One is you need a data related to cost, that I told you there are a sources from where you can get the cost data. Another things, because in cost minimizations only the cost data is required and you can perform a cost minimizations, but to do a cost effectiveness analysis you need the data related to outcome, and outcome is not only in terms of safety, it's also in terms of efficacy. So both data analysis is required for that particular delivery systems or particular formulations, whatever it is. So how to do it? How? So first is cost data you can get from a government websites, but you need the analysis of safety and efficacy data. So one of the if you go for a publication, you will get researcher have used a clinical trials data of the patients where they have used it. And most important tool is meta-analysis of clinical trial data is recommended for use of health economic analysis. So meta-analysis of clinical trial data means you are doing the analysis of 5-6 clinical trials related to that particular delivery systems in that particular disease. And then you will come up, okay, on the basis of these data, this medicine is that much effective. So that is a one part. And then similar way, you have to do the safety analysis. And then you have a data of cost. Then you can compare, okay, the treatment A is better, or this delivery system is better in, or this one is better in terms of cost and safety and benefit. So meta-analysis of clinical trial data you have to do. It's just an overview meta-analysis. I think maybe many of you knows about it. the systematic review. First, to do a meta-analysis, you have to do the systematic review of the literature. And systematic review of the literature, you have to be follow some steps. You have to be clarify your aim and objective, what you want to do. You have to be fit your inclusion and exclusion criteria. You have to do the quality assessment, and then you have to be extract the data, and that extracted data you can analyze by using these softwares, or you can do manually also, but software will make easy to calculate overall estimate. So different steps you have to be follow. And first, of course, you have to be search the databases. You have to search the clinical trial websites where you will get that informations. Okay, this delivery system, if suppose liposomal of that particular medicine, so you will get how much it's effective, how many, what was the sample size, how many patients receive it, and with respect to comparator, how much effect you got. So all these, first you have to be identify the studies by using these databases, and search you have to do in a proper way, in a standard way. It's not simply putting in a PubMed just cancer novel drug delivery system in the treatment of cancer. No, you have to make a proper search items. You have to make, okay, this is the liposomal of this one, then you have to think of what is the class of this one, then you have to be use and or techniques in the advanced search in the PubMed. And you have to be convert your research questions in terms of PICO format. PICO means you have to be define your patients. If it's a cancer patient, so which types of cancer patients, in types of cancer patients, which stages of cancer patients you are going to include, and what was the intervention. I means intervention, what was the intervention, it's a liposomal form or any form or whatever it is. And then you should define your comparator. You want to compare your delivery systems with which one. Are you going to compare with the conventional drug delivery system? Are you compare with the any other delivery systems? And then finally you have to define O stands for outcome. You have to define the outcomes in terms of efficacy. What is your primary endpoints? In terms of safety, what is your endpoints? So everything you have to be defined, and you have to do these analysis or searching of these studies as per the Prisma guidelines. You can get a more details on Prisma. And then after selecting the studies, suppose you have selected, you got 5 or 6 or 10 studies related to your delivery systems in that particular cancer. So next, what is the next? Next is you have to be do the quality assessment of the studies which you have included in your studies. So quality assessment, we have a scales you have to be used, and different scale. If you are included a RCT, then you can go with the consort guidelines. You can use the straw. There are so many scales, Newcastle, Ottawa scales. But only things you have to be careful when you are using this scale, you have to be see, okay, whatever scale I am using, this scale is applicable for my studies or not. Because design of studies are different. Some studies are RCT, some studies are observational, some studies in observational there is some are case control, some are cohort study. So accordingly scale has been designed, and accordingly you have to be select the scale to do the quality assessment. That is the next step. So after quality assessment, you have to be make a Excel sheet where you have to be added the informations. As I told, what was the total patients? If suppose 100 cancer patients, so out of 100, how many have used these liposomal drug delivery systems of this particular drug? And how many have not? How many are in a conventional drug delivery system? And out of 100, how many have received the response, how many have received that particular endpoints? So every information you have to be structured in a Excel sheets, and then you have to do the analysis of data. And by this Revman is a freely available, you all can install in your laptop. It will just ask you academic versions or your system configurations. That's it. You can use, everyone can use Revman. It's a freely in a non-Cochrane mode. And then by using the Revman, you can calculate the effect size, overall estimate in terms of odd ratio, relative risk, or you can calculate variance. It depends upon the what kind of presented with a 95% confidence interval. And there are two plots which we are talking when we are talking about clinical trial data analysis: forest plot and funnel plot. So I'm just going to give you overview what actually these plots. Maybe many of you have seen in research papers these kind of plots. So these plots is nothing but just a overall estimate measure. So you can see, suppose this analysis have contained 1, 2, 3, 4, 5, 6, 7, 7 studies. Maybe these are randomized clinical trials or observational studies. And these are the total number of patients, and out of total, how many have got the response, and in this is the comparator group. So this value is tell you about the odd ratio of individual studies. And in forest plot, only things which you have to be see is this diamond. This diamond will tell you about the overall estimate values. This diamond means these all studies data has been pulled, and this is the final concluded odd ratio of all these studies. This is 0.86 and confidence interval is 0.41 to 1.81. So it indicates there is a change, but change is non-significant because it crosses the 1 and our value is 0.86. So it says non-significant changes. So another thing is what you have to see in the forest plot is these values, heterogeneity values. So in the heterogeneities, you can see chi-square values, you can see the I-square statistics, where you can see 76%. So what does it indicate? It indicates variation among studies. Variation among study means you have included 8 studies, you have do the analysis of 8 studies. So how much variations among these studies are. So more the variations, less will be the confidence on your conclusions of that particular analysis, because more heterogeneity means these all studies have a different design, different sample size, different age group. So in that case, to verify the heterogeneity, you have to be perform the subgroup analysis. You have to be see, okay, out this particular drug delivery system is effective in how much effective in male, how much effective in female, and you have to be do the subgroup analysis in different age group, and so on. So this is you can get. And another plot when we are doing the analysis of clinical trial data of any whether it is cancer patients or any, you have to be present the funnel plot. This is a funnel plot which tell you about the publication bias. So in publication bias in funnel plot, if your funnel plot is symmetrical in nature, so that means there is a less involvement of publication bias. And if it's not symmetrical, you have to conclude that there is an involvement of publication bias. And this funnel plot will tell you only about the qualitative analysis of publication bias. To do a quantitative analysis, you have to perform other tests like Bagger test. These kind of tests you have to be do, where you can get the quantitative informations of publication bias. But funnel plot will tell you only the qualitative analysis. These are the recent publications which our group have done recently. One of the publication on cancer, where we have calculated in collaboration with Rajiv Gandhi Cancer Institute, Rohini, Delhi, where we have just calculated the prevalence of anemia among chronic myeloid leukemia patients treated with imatinib, a evidence-based meta-analysis where we get to know about how much anemia in the particular patients who are using a imatinib. So we got a significant correlations between these, and this is recently published. Other studies of analysis we are also published recently in European Journal of Pharmacology, International Journal of Clinical Practice, and ACS Chemical Neurosciences. So that is a one thing where you can get the, and these analysis not only for the efficacy but also for the safety you can do in a similar way. So as I told you, to do these analysis, what is required? You need an safety data, you need efficacy data analysis, and cost, right? So you can get these results from these studies. And cost data you can get from National Pharmaceutical Pricing Authority, Department of Pharmaceuticals, Government of India. Just you can go through these websites and PPA, where you can get the cost of the medicines. In then utility, you can click on compendium of prices and you can get the list. So I have just recently, one of my student is working on these pharmacoeconomic studies of rituximab or trastuzumab. So yesterday the cost of these rituximab is mentioned about 703, and this trastuzumab is 55,812. So now we want to compare, okay, whether this is that much money is worth to pay, or in terms of safety or efficacy, or we can go other kind of medicines. So next thing is, this is the one step which we have to perform. Next is, you have to be create a models, that is we call cost effectiveness model. And when we are talking about, it's very important, at this stage we have a data from a clinical trials. Our particular formulation is 1 year old, so you need something which will predict the future, which will say, okay, when you are using this particular formulations for 5 years or 10 years, what will be the cost, what will be the benefits, what will be the harms to you. So from where you will get that information? So you will get from creating a models by using the computational techniques, where you can predict the future, okay, by using these efficacy data from clinical trials which is available at time. You can make a models with the help of software, and then you can make a conclusion, okay, when we are using this for this year, 5 years, 10 years, so this will be the benefit, this will be the risk, and this will be the that much cost you have to be pay for that one. So cost effectiveness model, so as I told you, model means you predict the cost and effectiveness of intervention based on the best available evidence. So best available evidence which you have today, on the basis of that one, you will predict the cost and effectiveness as well as the safety also. So it provides a robust analytical approach for policymakers and regulator to assess if the cost associated with an intervention is worth paying or it's not a worth paying. So model which we are using is well-known model is Markov models. Many research publications, you all can see Markov models. So Markov model we are prefer when we are talking about a chronic disease. When we are talking about a acute disease with no future impact, we go with the decision tree models. There is a complex disease like cancer, we go with the micro simulations. Even you will see Markov model also in cancer papers also. Transmission modeling we are doing for a communicable disease or vaccinations. So these are the different models. So just give you an idea what actually these models, how we are doing. And by using the MS Excel also you can do these models, but of course it will take little with much more time and more effort. So what is the concept of these models is, the study disease we are dividing into a two distinct and mutually exclusive state, and then patient can only be in a one state at a time. Then we are assigning a transition probability are assigned to represent the patient moving between these state over discrete time period, that we called a Markov cycles, and apply of these transition in model and attaching estimate of resource use and health consequences to the state, right? And running of model over a large number of cycles, then we will get to know. So I have just added one example, these models, because before you, as I told you, you can do by using the Excel also, but before doing it or using the software, you have to make a sketch on your paper, what actually, how, what your model will be, what are the different stages, what how you can differentiate. So in this cancer I have just included, there are two possibility: patients will remain undetected and patients will detected. So the patients will undetected and there is a chances to go into the death, and detected patients there is a chances of recovery, and detected patients can be fall into a following categories. It can, there is no need of hospitalizations if it's a early detections, but it's very difficult in cancer. But if it is, then there is no need of hospitalization, only by doing the OPDs or using the medicines the patients can be treated. There is a need of hospitalizations, there is a need of hospitalization plus ICU, there might be need of, there are some patients, some percentage of patients need hospitalization plus ICU plus ventilators, and then patients can be died or can be recovered. There are two options: patient can be died or recovered. So by that way, this is a simple example I have added. So you have to think about what are the possibilities if person is having a cancer, so what are the possibilities, and accordingly you can create a models, and then you use your whatever delivery system or whatever drugs you are using. But limitations of cost effectiveness analysis is different outcomes you cannot be compared. Like if you want to compare prothrombin time versus blood glucose, you can't compare. Your primary endpoint will be same, your formulation can be different, you are taking liposomal or another delivery system, you can compare, but your primary endpoints should be same. Like different category of classes of drugs also you can't compare, like diphenhydramine with fexofenadines, maybe other class you are comparing, no. that is also the one of the limitations. So quickly, I think time is okay. So quickly, I am going through what are the different steps when if you any one of you want to do pharmacoeconomic studies in cancer or any disease. First step is you have to define the problem, state the problem, select the objective, as whether you want to compare new drug delivery system with conventional drug delivery systems, or you want to compare different novel drug delivery system, whatever. So you have to be define your problems, and then you have to be define the study perspective. You are going to conduct studies in perspective of patients, institutional societies, or regulators, whatever. So you have to define it and identify the alternate intervention and outcomes. You have to be identify, and then you have to say what are the alternate interventions for this particular disease or in this particular area. Then you have to be select the appropriate pharmacoeconomic method. So as I told, in cost minimization method is not appropriate if outcomes are not equal. If you are not sure about outcomes, so you can't go with the cost minimization. You can go with the cost minimization, then you are assuming are equal. The best example is comparing generic benefit analysis. Outcomes, we are measuring in a monetary terms in dollars. Cost effectiveness outcome measure in a natural terms like morbidity, mortality, and cost utility. We go with the quality adjusted life year. Then fifth step is you have to be select the primary data source and decision from where you will take the data. So you can take from a retrospective studies, you can date from a prospective study, clinical trial, economical trial, databases, meta-analysis. So all these you have to define from where you are taking these data. And then you have to be select the analytical techniques, which models you would like to use, whether you are going with the Markov, Monte Carlo simulations, or decision tree analysis, whatever. Establish, next step is you have to be establish the probability of outcome event. And how to calculate the probability of outcome event? It will calculate by using the hospital record, efficacy rate, incidence of adverse drug reaction, prospective study if needed, as well as the meta-analysis data is very helpful to calculate the probability of the outcome event. Then you can make a decision tree, and then you can see, okay, this is my decision tree and these are the probability, and then you can calculate the ICR. Then next step, you have to perform the sensitivity analysis. Sensitivity analysis means by changing some variables, suppose you have to assume 2-3% of discount or something. So by changing that variables, your conclusion should not be effective. If your conclusion is affected, so you have to mention. That's why sensitivity analysis is important. You have to perform the incremental cost analysis again by doing the variations, and then you have to see the impact on the conclusion. And finally, you have to be present the results. You have to describe the assumption, but method you have used, what are the data sources. Data sources are very important, because if your data source is not authenticate source, so your analysis result is not valid. Critical variables, you have to do the sensitivity analysis, study limitations, everything you have to be mentions in the results. And these are the commonly used software which we are using in a pharmacoeconomic studies. So you can use it. TreeAce we are using, but of course it's a commercial one. I think it's around 1 lakh cost, but it's freely trial is available for 14 days. So 14 days trials any students can use, but before using it, first learn how to do these analysis and then install it, then use it. Otherwise your machine will registers, you can't install it again and again. Freely available tools are also available, and these are WHO choice tools where you can perform these cost effectiveness or these modeling, where you can create a model. So these also very useful tool which you all can assess it. And I think everyone knows about these societies. It is International Society for Pharmacoeconomic Outcome and Research with respect to pharmacoeconomic studies, where you will get a more updates upon these research. Career opportunity for students, if you have these skills in these particular areas, there are a lot of opportunities with good packages. A few of CRO I have mentioned, breastmed in Gurgaon is purely doing the research on these health economics, it's pharmacoeconomics, and providing good packages to the students. WNS Novartis is doing these economic study, Parexels, and so many CRO as well as pharmaceutical companies, because these are very important studies. Every company want to know whether this treatment is going to be cost effective or not. So you should know about it. And if someone want to do a research or PhDs or further research on this area, so of course researcher want to publish in a journal, so where you can publish what are the, where is the dedicated journals. So dedicated journals elsewhere, value in health, expert review of pharmacoeconomic and outcome research of Taylor and Francis. There is a Springer journal dedicated to pharmacoeconomic. JAMA have a section for a pharmacoeconomics where you can see these research studies and you can submit to these dedicated journals with respect to pharmacoeconomics. So conclusion is, it's not a simple analysis. It looks like many students think about, okay, pharmacoeconomic means की cost analysis. ओनली आपने एक कॉस्ट को प्लस कर लिया कि इतने इसकी 5 रुपए की है, ये 10 रुपए का पैकेट पड़ रहा है. इट्स नॉट ए दैट analysis. You have to be consider safety as well as efficacy analysis. Then you have to be conclude that whether this is cost effective or not. And when we are doing these studies, we have to be follow some checklist, and checklist, well-known checklist which we are using is Consolidated Health Economics Evaluation Reporting Standard Checklist, that is CHEERS. Many factors must be addressed and careful planning is required. You have to think about, and finally you can submit your finding to the policy makers also, and it will help to make any regulations on these particular medicine. Expert opinion is required. Proper application of pharmacoeconomics will allow the pharmacy practitioner, health policy maker to make a better and more informed decisions. So with these words, I would like to thank MIT University again, Quelliers, all the management for giving me these opportunities. And you can assess these. We have some websites where we have updating time to time what kind of research we are doing and what outcomes, what findings we are getting. So thank you so much for listening me. Thank you. Over to you, sir. Any questions? I am happy to answer.
Now is the time to interact with the guest speaker. If any student or any other participant having any query, you can come forward and ask the questions. Anyone having any questions? You can come forward and ask the question.
Any student, any question?
If anyone has any question, can write in chat box
Student,
also.
any have question?
So I can
Shubham.
read from chat box.
Hello.
Yes.
What is the disadvantages of novel drug release system, and which method is more effective?
Okay, so novel drug delivery system have various advantages, but of course every system has a limitations also. So if we are talking about monoclonal antibodies, so we have seen some allergic reactions has been observed with respect to that particular system. With some system there is a biocompatibility problems. With some systems, again, it depends upon the nature of your molecule. Your molecule is depending upon your nature of molecule, you decide which drug delivery system is best for it. But of course, again, means with doing all these things, again, we are not able to minimize adverse drug reactions completely. Still, we are getting some adverse drug reactions. So means these are not very much specific to that particular target. But of course, when we compare with conventional drug delivery system, these are more safer as compared to conventional. And another questions you have asked, what is your second part?
Good morning, sir.
Good morning. Very good morning.
Sir, my question is cancer genetic?
Yes. See, etiology of cancer is still unknown. We are assuming various environmental factors. When these environmental factors are interacting with the genetic factors, it results in the development of various cancer. So, but again, it doesn't means if someone have that particular gene, so that particular child of that parents will definitely get a cancer. It depends upon luck also. Sometimes that will modify and result in the development of cancer. But nowadays, why cases of cancer is increasing year by year is there are a couple of reasons. It and the major reason is environmental factors, pollution levels, pesticide use, when you heavy metals in drinking waters, vegetables. There are a couple of reasons for it, but exact etiology is still unknown. But we are assuming these are the factors which results in the development.
If it is genetic, so is there any method to cure it?
Yes, genetic. When in with respect to genetics in animal research, we are doing knockout mice where we are particularly deleting that particular gene, and then we are seeing whether it's going to cure. But still, we don't have that particular medicines with respect to gene therapy in the market, as far as I know. And whatever the gene therapies are available, or when we are talking about a personalized medicines or anything, these practically it's very difficult to implement in a practice, particularly in country like India, because cost is very high, because you have to do the sequencing of every patient sample, so it's very difficult to do it. But of course, when we will get to know this particular gene is involved in this particular cancer, so we can develop and research is going on in this area.
Okay, thank you so much, sir.
Very nice, thank you.
Any student?
So my question is,
Yes.
when should I get tested for cancer, sir?
Okay, very good questions. Because symptoms, on the basis of symptoms, it's very difficult to diagnose whether the person have a cancer or not. But whenever any unusual things in your body you are feeling it's a unusual, I have not experienced this earlier. Of course, you have to go for a various diagnostic test. In diagnostic test, when you will go for a, you will rule out for other disease also, not for cancer. But when treatment is started by diagnosing that particular disease and still you are not getting effect, then physicians should verify for a cancer also. They will look for your blood profile, they will look for your other possible reasons that might be. But if you ask me when you will go for a test for a cancer, no need to go for a testing of cancer unnecessarily. You have to go when something unusual you are feeling in the body, then of course that time you have to go for all the diagnostic tests, right?
Okay, thank you so much, sir. It was wonderful knowing your views and opinions on pharmacoeconomic analysis and cost in the reduction of cancer burden. Proper application of pharmacoeconomic will allow the pharmacy practitioner and administrators to make better and more informed decisions regarding the products and services. Sir, kindly accept our virtual felicitation for sharing your expertise in the session.
Thank you so much. Thank you so much. Thank you.
Thank you so much, sir.
Thank you so
Now,
much.
moving to the second session of the conference, I would like to invite Dr. Satish Shilpi for a brief introduction of our next guest speaker, Dr. Sharat Prakash Pandey. Sir, please.
A very good afternoon to all. I am Dr. Satish Shilpi, and on the behalf of AIP, Amit University Gwalior, I welcome you all in the session second of day first of this e-conference entitled Novel Drug Delivery Strategy for the Treatment of Cancer. So, and it is my great pleasure to introduce. It is my great pleasure to introduce our guest speaker, Dr. Sharat Prakash Pandey, Faculty Department of Pharmaceutics, Sri GS Institute of Technology and Science, Indore. He holds a doctoral degree in pharmaceutical sciences from RGPB, Bhopal. He qualified for the GATE 2004 and completed his post-graduation from SGITS, Indore. After completing his master's, he served He served the Truba Institute of Pharmacy, Bhopal in the capacity of associate professor and academic in charge. He is a non-educationist and researcher with 12 quality book chapters in different books and more than 30 publications in reputed in national and international journals. He has filed 3 patents and co-investigated the research project funded by the Madhya Pradesh Council of Science and Technology, Bhopal. He developed the 3E content through the National Mission on Education. It is an initiative of MHRD. He has been the recipient of the Best Faculty Award in 2012 and Innovation Award for Scientific Research in 2018. He is a Vice President of Society of Pharmaceutical Education and Research, MP State, Executive Member of IPGA, Madhya Pradesh, and member of IQAC, People's University, Madhya Pradesh. Along with this, he is currently a member of different bodies such as APTI, IPGA, SPER, and the Society of Ethnopharmacology. Now, I request Dr. Sarath Prakash Pandey to take over the session. Sir, please.
Thank you, Dr. Silpi, for such a nice introduction. Please just give me 1 minute to share my screen and then I'll start. I think I'm audible to everyone.
Yes, sir, you're audible, sir.
Yeah, thank you, sir. So please, sir, just confirm that my screen is visible to everyone.
Yes, sir, it is visible, sir. Make it full screen. Thank you, sir.
Thank you, thank you. So first, again, just I'm very thankful to Dr. Satish Silpi, sir, for such a nice introduction. And before moving ahead, just I would like to thank the management of MIT Institute of Pharmacy, the Vice Chancellor of MIT University, Dr. Neeraj Mishra, sir, and all the staff members involved in this e-conference session to give me an opportunity to interact with such a budding pharmacist. Before moving ahead, just I would like to confess one thing. This is a little bit a critical situation for me right now, because I think everyone will agree with this fact that if you are presenting any talk in front of your teacher, so that is going to be difficult for you. And very few of you are knowing that Dr. Neeraj Mishra, sir, was my teacher, my mentor. He taught me in my UG. And continuously, he's just supporting me, he's guiding me. So definitely, it's a little bit, just you can say, hesitating situation for me. But at the same time, as I told you, it's a confusing one. I'm also a little bit confident. Because whenever just you will feel any problem in your life, your teacher is there. So definitely in this session, I'm very much confident, because if I will make any mistake, any sort of mistake will be there, that definitely my teacher, my mentor is there to support me. He's in the backup. So thank you, sir, Mishra sir, for guiding me continuously. With your permission, sir, I think just I can start now. So, yes. My today's topic is role and significance of nanocare used for the development of theranostics in the management of cancer. Now, the basic question arises. Suppose if we are going for anything, any talk, or suppose just we are preparing any papers, always there should be some specific motive. So why just we have chosen that? Number of diseases may be there, number of problems may be there, but this is the topic which we have chosen here. We have chosen for your conference. The reason is very simple. Everyone is knowing this fact that cancer is one of the leading death cause in the entire world, and it is affecting the entire world irrespective of its demographic and economic variation. I'm not going in very detail, because everyone is knowing what is cancer, how it is getting grown and all that. It is clear to everyone that if pro-oncogenic system is getting aggravated and your tumor suppressive system is getting deactivated or it is working less, in that situation the imbalance may lead to the cancer. Number of reasons may be there. It may be any drug, or it may be any sort of radiation, or it may be any genetic variation. Anything may be there. Because of that, someone may have the cancer. And once someone get infected with this one, now the life is getting a little bit difficult for them. The reason, mortality is very high. The second thing is that suppose if we want to cure that, I think everyone will agree with this one, whatever the drug we are currently using, all the chemotherapeutic agents, they are highly toxic. And I think in the late 90s, it was very famous, especially in the journal papers, in the layman papers, that either just you will die with the cancer or you will die with the your drug. Because of cytotoxic behavior. As a pharmacist, if I say, drugs are going to save you, there will be no problem at all like that, someone will die from any drug. But definitely the afterlife of any cancer patient was going to be very difficult because of the toxic impact of such medications. That was the biggest issue. The second problem which is currently happening, that is the cancer diagnosis. Our diagnosis of any disease, not only in the cancer. If we are in a situation to diagnose any disease in its preliminary state, then definitely we can control it, we can manage it in better way. Especially in case of cancer, because again everyone will admit that after the second or the third stage, once the metastasis start, it is very difficult to control the situation. While if we find it out, yes, cancer is there and we recognize it, we diagnose it in the early stages like the stage 1, stage 2, then it will be more easy for us to control. We have witnessed number of advancement. Significant advancements are there, but still we are lacking little bit. And currently, as I already told, the number of diagnostic techniques are there: MRI, computer tomography scan, positron emission tomography, that is a PET scanning, which is very common, photoacoustic imaging, and number of other techniques are also there, which can be utilized for these purposes. But at every time, some questions also arises regarding the spatial resolution, regarding the sensitivity, regarding the specificity, because these techniques are going to use some sort of contrast agent. How this contrast agent is behaving in the physiological system, that is going to be very important. Now, number of cancers are there. Number of cancers, I think in the morning session, Dr. Vipul Sharma was there who was telling, and I was very overwhelmed to see his knowledge about the cancer and about the pharmacy and novel delivery system. He has told a number of cancer. I'm just taking one example. Suppose breast cancer is there, lung cancer is there. Yes, we can control that in a way. Suppose just we have go for the surgical remover, but I don't think so. With that is the clear-cut management therapy. Because if any single cell will be remain there, then again the chances of growth will be there. Second thing is that once we gone for the removal, then what will happen? As per the cosmetic abstract, it is not going to be appear very good. What should we do? How just we can manage that? These are a number of questions. Apart from that, I think everyone again will agree with these facts. The majority of drug, whatever we are using currently for the management of cancer, they are hydrophobic in nature. And that is the biggest problem with regard to their viability. These drugs have the less viability. Toxicity associated with these drugs start from the therapy till date we are facing it. Then another issue what we are currently facing, that is resistance for the therapy. Suppose docetaxel has been started to serve a particular patient, but after 4 to 5 times might be possible that drugs may not respond. That could be just because of the resistance. Now, just we have to understand why this resistance is occurring. Anytime, whenever just we will go for the cancer chemotherapy, and suppose we are giving any drug, immediately your cell start to develop P-glycoproteins, and overexpression of these P-glycoproteins may be reason for that, because these glycoproteins are pumping out the drugs from the cell, and once they get overexpressed, now the drug is not going to be active anymore. So just we have to control the situation too. These are the hurdles. These are the barriers if we want to develop something, if we want to cure the cancer in a more effective way. Apart from that, already I had discussed the problem associated with the diagnosis is again a big issue. So as a pharmacist, what we did and what we are doing, because every time the society is going to ask us what we did and what we are doing. So what we did, initially just we'll start to think about the characteristic of drug, the problem associated with this one, that is the bioavailability, then the toxicity associated, everything we will consider, and how just we can do that. What we have done in the previous time, just we have developed some nano carriers. And these nano carriers have solved almost these issues, because hydrophobic drug can be just administered after incorporating into the nano carrier system, and that has almost finished the problem related to viability, or just it has minimized as such. Second thing is that the ready toxicity get minimized. Third thing, once you incorporate something in a nanocarrier system, now the PGP receptor, PGP proteins are just not recognizing that drug, and because of that, the resistance was also minimized. Similarly, already I had told, targeting was a big issue, because once you are giving contrast agent as such, so it is just roaming in all over the body system. So by using the nanocarrier system, what we started to do, just we have incorporated the contrast agent in the nanocarrier system, and these nanocarriers get located to the specific position that was the tumor or in. So the first thing, whatever we have done, that is formation of nanocarrier. Number of things may be there, number of things. It may be polymeric nanocarrier, that is polymer nanoparticles, muscles, polyplexes, tendrimers, or it may be lipid-based nanocarriers, liposomes, neosomes, nanomodulation system, lipoplexes, SLN, and so on. Apart from that, just we have developed number of inorganic nanoparticles which were more effective especially in case of diagnosis. Iron nanoparticles, gold nanoparticles, silver nanoparticles, quantum dots, carbon nanotubes, and so on. That was not very much effective even. Why this is so? Because we have reduced the size, we have increased the bioavailability, and we have utilized to the inherent characteristic of tumor. What was the inherent character? The simple thing is that in case of tumor, what happens? The vasculatures get increased, and these vasculatures are faulty, they are leaky in nature. And because of that, if vasculatures are leaky, so if minor or tiny particles will be there, they are going to cross this one. Yes, it is something like that. If we have prepared something in form of nano carrier, whenever just I will utilize that. Just 1 minute. Yes. What was happening? Whenever it was moving in the normal cells, at the tight junctions are there, it was not moving. But in case of your tumor cells, as leaky vesicles were over there, and one thing which start, that is called EPR, enhanced permeability retention. That was the major factor which was leading to the accumulation of these nanocarriers in the tumor system, and we were getting the effect of our drug. But that was a little bit difficult. The reason was very simple. Whenever you will administer something to someone, immediately your RES system get activated. Means your immune system will start to work against that. Macrophages will be there. And as you will administer it may be anything. It may be liposome, it may be SLNR, anything. Your body is going to recognize it as a foreign particle. And as it will recognize it as a foreign particle, definitely they will start to counter it. So RES will start to engulf it. And once RES engulfment will be there, then again your formulation is going to be finished. As such, everyone is doing this one. Two ways may be there by which we can bypass this RES. The first one was the inverse targeting technique. In the inverse targeting technique, what we were doing, we were taking dextran sulfate injections. And a pre-injection can be given to someone, and because of that reason your RES get inactivated, and now you can administer your formulation as such, and there will be no problem at all. And the second thing was that if we can make our formulation long circulating, we can dodge this RES. How just we can do that? Just by stealthing it. How just we can do that? Obviously, the best approach was to use PEG. As just we provide hydrophilicity to our system in form of PEG, we can prepare the pegylated liposomes, pegylated SLNs, and so on. So because of that, what was happening? The RES was not recognizing that, and because of that reason, your formulation, whatever the nanocarrier you were having, that was long circulating. And when it will be remaining in your circulation for the longer time, because of passive targeting, whenever it leads to the tumor region, it is going to be accumulated, and that was much easier. No problem was there. But still, now just I will say, think about a situation. Suppose if you have written a letter to someone, that is a simple paper, you have written something. A paper you have taken and you have written something. What is the situation? It is not the safer way. Anyone can hear that. Yes, suppose water is there, but definitely this may be spoiled. What we do, just we put it in envelope to protect this, the initial paper. In this phase, just we are doing the same. We have taken the drug and we are putting that in an envelope-like situation. So just we are protecting it. We are increasing the efficiency of this thing. Second thing, envelope was not sufficient because again, still water may affect this one. So another wrapping just we can do, that may be a plastic sheet. Just we can take a polythene. We are providing the additional security to our system. This RES, just we can consider as a plastic one, which was providing the more security to our system. We were protecting our system from our body as such. But do you think, is it sufficient? Suppose if you have written your letter to your parents, is it possible to reach it to them? No, because you haven't put their name. Until unless you put the address, name, and everything, it is not going to reach the destination. And similarly, just we have to think here. We have to provide the destination. We have to give the address to these envelopes. What we started to do, just we have started to utilize, just we have gone for the functionalization of these nanocarriers. During the functionalization, what we were doing, just we were taking the different macromolecules, or just we were utilizing the specific character of any specific tumor. Like suppose a tumor is there and it is overexpressing to the transferrin receptors, or it is overexpressing to the folate receptors. What we will do, just we will tag our formulation, our nanocarrier with folic acid as such, or with the transferrin as such, or any other peptide, or any other aptamer may be there. And that will serve as a address on your envelope. Whenever just you will put it here, you will functionalize your nano carrier. Now what will happen? It will start to move in your circulation, and wherever the tumor will be there, which is overexpressing to you these receptors, it may be folic acid, transferrin, or whatever, immediately it is going to bind there, and internalization of these carriers will be there, and it will release the drug, and you are going to get the results. But was it enough? Once you have functionalized, obviously it is getting binded more efficiently to the cancerous cells rather than the normal cells. In case of normal cells, has no addresses there, so it is not going to accept as such. But was it sufficient? As a pharmacist, I think it's no. The reason we are continuously thinking about the therapy part only. While every time in the initial just I had told, diagnosis is very important. And it's very crucial, especially in case of the cancer. If we can diagnose the disease in the early stages, then we can manage it more efficiently. So, in the another step, the scientists have started to develop some diagnostic agent based on these nanocarriers, and by virtue of functionalization, they have increased the efficiency of contrast agent. Just they have increased the sensitivity of currently used diagnostic methodology. And it seems, yes, now we have done everything. We have developed the therapy. We have just gone for the diagnostic part. But still, every time these are the different parts, and every time just if you have to go for the diagnosis, you need another time for the therapy. Again, you need to go for the little bit. It is going to be cumbersome for a patient. So again, the scientists started to think how just we can go for the better patient compliance, how just we can minimize the problems associated with the therapy and diagnosis. So currently, the majority of pharma fraternity is working on the theranostics. Theranostics means just we have combined the therapy and diagnosis as such. Suppose if I am having a liposomal system, I will have doxorubicin there, and at the same time, just we can also include quantum dots into that. So in the real time, just we can locate where, because these quantum dots are going to have some fluorescent action, and easily it can be detected. So we can find out where my formulation is going. In the real time, just we can detect that. First thing. Second thing is that in the real time, suppose if you have to go for the surgery, by locating these, just you can locate a specific location where just we have to go for the surgery. So by including different diagnostic materials, different contrast agent and drug in a single system, we had made the things more easily, more comfortable. Still, there is no FDA approved. Again, just I will make it clear to everyone. No FDA approved theranostics are there. But still, we are working vigorously. Everyone is working, and we are expecting that in the near future, definitely we will have some approved theranostic, because number of formulations are there which is already in the clinical trials. And we accept, because when this formulation has been utilized in animal models, they were having the super results. So one by one in the coming PPTs, just we will discuss on that. And that was the destiny. This is the thing what we want to do. We want to reduce the toxicity related to drug. We want to make it more targeted. Wherever I want, just I will target the drug only. These are the various ways by which just we can modify the surface of nanocarriers. So initially, just I will talk with the transference. Just a minute. 1 minute, please. So in case of transference are basically 78 to 80 kilodaltons molecule monomeric glycoprotein, the primary role. Serum iron transportation. I think everyone was doing this one. Every single cell is going to have the transferrin receptor which is responsible for RNNT, which is necessary. But the study shows that TRF, that is a transferrin expression, get increased up to 100 folds, up to 100 folds. And at the same time, when the cell is getting divided, as in case of the cancer, the iron requirement also get increased. So at one hand, the iron requirement get increased, and at the second hand, in our cell, in our system, expression of these transferrin system get increased. Now just we can say, yes, it can be easily located to where the tumor cell is there. Now suppose if I'm having any nanocarrier and if I tag it with the transferrin system, wherever a tumor will be there, which is expressing to this transferrin receptor, that is going to bind that position. And further targeting can be achieved. Here just we have one example where someone has prepared doxorubicin loaded PLGA nanoparticle, and then further just they have coded with the transferrin system, and they have got excellent results. Even it has been observed, again, everyone knows, blood-brain barrier is highly specialized and it is very difficult to cross that. But in case of those nanocarriers which were decorated with the transferrin, it has been observed that they were crossing easily to the blood-brain barrier, and by using these formulation, even we can control the disease related to your brain. If brain tumor is there, again, you can utilize these formulation as such. The second one, which is very important one and easy to prepare, that is to decorate your nanocarriers with the folic acid. Folic acid is easily available and everyone knows that it's a molecule, and being non-immunogenic in nature, because immunogenicity in any formulation may be a bigger issue. It may be a bigger issue. So if something is non-immunogenic, then definitely you can utilize it as such. Plus, folic acid is required for the optimum health. Again, everyone is doing that. It is a cofactor of various enzymes, especially which are involved in the DNA and RNA synthesis. And again, during the suppose the cell is getting dividing so fast. So what will happen? The DNA replication is going to be there. That means the consumption of folic acid by a specific position, specific tissue, specific cell is going to be increased. And we can utilize that. If a cell is demanding the folic acid, yes, we will give it. But with the folic acid, we will also attach our nanocarrier, which will already have a drug. Folate receptors are glycosyl phosphatidylinositol anchored cellular receptors, and it is abundantly expressed in different cells, different tumor tissue. In the normal condition, in the normal cells, again you may have the folate receptor, but they are not the overexpressed. In different cancers, epithelial, ovarian, cervical, breast cancer, lung cancer, kidney cancer, any of the cancer may be there. In each and every conditions, the chances of overexpression of folate receptors are going to be very high. So we can utilize this folic acid, just we can functionalize our nanocarrier by using this folic acid, and it is comparatively in comparison to the others, like transferrin or like the aptamers. They are going to be just, you can say, the lower cost is getting involved with that. So we can utilize it more in a more easier way. And we can target our tissues. Then the another one which we can utilize for the functionalization of a nanocarriers are aptamers. Aptamers are basically unique three-dimensional structures having single standard oligonucleotides, which exhibits a specific binding to the homologous type. It may be anything. It may be protein. It may be any specific cell surface. It may be a tissue surface. Plus, in comparison to antibodies, these are comparatively small. So it will be more easy to carry your nanocarrier system. They are less complex. They are flexible in nature. Plus, lower immunogenicity. Again, it is going to be very important because of its nature. And it can bind to their specific target with high affinity. So these are the basic characteristics which makes it more more useful for the targeting purposes, for the functionalization purposes. And these aptamers have the high, just you can say, the capability of identifying the minute variations. Suppose a dividing cancer cell is there. So obviously, some sort of chemicals are going to be released, some sort of newer proteins may get formed. Or if benign tumor is there, so definitely the environment is going to be a little bit different. In case of malignant tumor, it is going to be different. So these minute variations again can be found out by the use of aptamer because it may identify them. So easily, if you want to distinguish between the malignant and benign tumor, you can do that with the help of aptamer. Number of aptamers are there. Here just are taken one example, two examples. That is the REG1, that especially may be also used as a drug for the anticoagulation. Apart from that, AS1411 aptamer is there, which is unmodified guanosine rich 26 mer DNA strand. By decorating your nanocarrier with such aptamers, apart from that, number of aptamers are there, which you can get and you can utilize as such for your developing your delivery system for targeting a cancerous cell system. Then another one is peptides and monoclonal antibodies. Again, we have studied a lot about these things. Almost we can put that because high diversity in the peptide sequence plus ability to identify the biomolecules, and as it may be a part of the body system, just biocompatibility is going to be higher. Again, these are the characteristics which makes the peptide very good ligand for the development of the delivery system. Number of things may be there, as already I had told, the environment of your system is going to be changed. What type of changes may be there? The first one is rapid growth of tumor endothelial cells may be there. Integrin, presence of integrins, because in inflamed cells plus in the dividing cells, the chances of development of integrins are going to be higher. So just we can find it out, and accordingly just we can decorate our nanocarriers with a specific peptide system. Apart from that, presence of aminopeptidases may be there. Tumor-associated macrophages related to lymph angiogenesis may be there. So number of factors may be there which can easily be located, which can easily be found out, and you can target with your specific peptides. High expression of epidermal growth factor receptors may be there. Transferrin receptors will be there. So accordingly, just we can take a specific peptide and just we can decode. Some examples are RGD, NGR, LIP1, CLT1, and so on. Currently, we are using the RGD and NGR. People are using very much. RGD is basically arginine, glycine, and that is a sequence of your amino acids. The 3 amino acids peptides are there, and it is a very good ligand, especially for if you want to target, if you want to modify your nanocarriers. Apart from that, number of monoclonal antibodies already just have studied that can be utilized. Plus, suppose HELA receptors are there in case of breast cancer. You can utilize these antibodies for decorating your for the functionalization of your nanocarriers if you want to target your breast cancer cell. Similarly, different antibodies can be taken and just it can be utilized for these purposes for targeting different tissue systems. Apart from that, some cell penetrating peptides are also there, just we can put that in peptide category. But here just we have taken as another class. CPPs are basically short peptides. They may have less than 30 residues, and based on their origin, just we can classify in 3 basic categories. First one, protein derived CPPs, chimeric CPPs, and synthetic CPPs. Protein derived CPPs are tag proteins and penetrating. These different penetrating peptides, as the name indicates, suppose if you are facing a problem related to the penetrability, if your carrier system is not able to penetrate any system, In that situation, that can be utilized and that may help to enter into the system. At the same time, it may also overcome the probability of drug resistance. In the initial, just I had told, P-glycoproteins may be there, that may be overexpressed in the cancer cell, that which is pumping out the drug from the cell system. So just you can overcome this situation too. If you conjugate these CPPs with the drug, then this problem can be overcome. Then these are some examples, as already I had mentioned, the number of formulations we can utilize, number of carriers we can utilize for the purpose. Here I will not go in the detail how the polymer, what a polymer nanoparticle is, what is polyplex. But here I will tell you how the people have started to utilize these things. Polymeric nanoparticles have shown their tremendous utility as a diagnostic and therapeutic especially. I think everyone is knowing about that, what is the polymeric nanoparticle. Suppose if we have polymeric matrix and the drug is entangled into that. Easily just we can load the drug, we can load the diagnostic agent and then we can utilize it. And even at the same time, we can just load drug and diagnostic agent as such. So we can use it as a theranostic, as a diagnostic, or separately we can use it for the therapy purposes. Then currently we are using, especially in case of the cancer, we are using the NIR dyes. And these dyes have number of limitations. Number of limitations related to their stability, their protein binding. These may be some sort of issue. Now, how just we can overcome? Suppose if we add these dyes in the polymeric nanocarrier system, now what will happen? The problem associated, the shortcomings of these IR dyes can be minimized and we can utilize it as a NIR proof for the diagnostic purposes. Number of polymeric nanoparticles have been generated and they are getting utilized for immunoassay purposes. Analysis of different type of biomarkers. Number of AGR may be there. So, number of biomarkers are there. Just you have to find out, and accordingly, just you have to find antigenic system, and you can find it out. Then, as a contrast system for imaging-based cancer diagnosis, already had 2 NRI dye can be loaded. Or suppose quantum dot is there. As such, quantum dot may be toxic, but whenever just you load it in a polymeric nanoparticles, it will be more easy to deliver it. Plus, if we functionalize these nanoparticles, it will be more easy to target at a specific location. At the same time, 2 different type of contrast agents can also be included in a single polymeric system, and that may be utilized for the multimodal imaging. You can just go for the MRI and SPECT as such. So, multimodal imaging is again possible with these two. Already I have mentioned the shortcomings of the NIR dyes can successfully manage with the development of several polymeric nanoparticles. Plus, suppose a nanoparticle is there and you have loaded the dye. Once you inject that, it starts to move in your circulation system, and where it gets deposited, if it is functionalized well, it will go and it will get deposited on the tumor system. So during the surgical removal, it may also work as a surgical guardian, because whenever you get the fluorescence, easily you can identify, yes, this is the location which have to remove. Plus, each and every polymeric nanoparticle have better surface to volume ratio. Surface functionalization is comparatively more easy. Already, just we have discussed that. Plus, size can be controlled. It can easily be tunable. If easily you can make it tunable, the simple thing is that the size can be easily controlled, and these are the various factors which make it more useful for the different users. Like if you want to detect the metabolite, you can go with that. You can go for the immunoassay. If you want to get the want to detect the toxins, you can utilize the fluoroimmunoassay and you can go with that. Number of papers you can find it out regarding these things. Even Bacterial cells can be find it out by utilizing these polymeric nanocarrier. Then another polymeric based system is polymeric micelles. In number of people, just you will find it out. The polymeric micelles are getting formed. Just people are preparing that for preparing polymeric nanoparticles. The ultimate purpose is to prepare polymeric nanoparticles. But as such, these polymeric micelles can also be utilized for such purposes. Polymeric micelles are self-assembled nano-scale colloidal aggregates of amphiphilic block polymer. Just you can simulate it with the like a surfactant system, which is having a hydrophobic part and a hydrophilic part. Hydrophilic and hydrophobic part. It is almost similar to that. Whenever just we go for the blocking, we can use a hydrophobic polymer and we can take a hydrophilic polymer, and with the simple reaction, just we can attack with these things, and they are known as block polymer, di-block, tri-block, different type of polymers may be there, and we can utilize that. Because if they are having a hydrophobic and hydrophilic group, whenever just we will disperse that in the water system, what will happen? They will start to form a micelle-like structure. They will start to accumulate in such a way so that hydrophobicity would be maintained for the longer time. And it almost get oriented in such a way. So suppose if I'm having a hydrophobic drug for the therapy, just it can be loaded somewhere here. Apart from that, suppose if I want to utilize it for a diagnostic purpose, then what can I do? Just I can take a metal chelate. Already everyone is knowing gadolinium is FDA approved diagnostic agent. So that's why just we have taken a example of this GDDTPA. This metal chelate as such is difficult to load that because it's a hydrophilic in nature. You can't load that in the center system. So what we can do? We can take a hydrophobic polymers like the phosphatidyl ethanolamine or the styrylamine. Once we make the block of these in form of this, again the system will start to work as a unit of your muscular system. Now what will happen? The hydrophobic part of your distillate system will combine with the hydrophobic part of your muscular unit, and the tail, which is having GDTPA, that can be just embedded into the hydrophilic tail system, and at the same time you can load your drug acid. So if you want to utilize it for the diagnostic purpose, if you want to utilize it for the therapeutic purpose, if you want to utilize it for the theranostic purpose, just you can go for that. But again, as just I have to, theoretically it seems much easier, but whenever we go for the practical, number of things may be there which may have the impact, number of variability may be there. So every time we need to optimize that. After the proper optimization, I think we will be able to prepare a better theranostic system. Then polyplexes may be there. Polyplexes are basically, they are just, we can utilize the cationic polymers and we can condense it with anion DNA. Means polymer plus your DNA or siRNA. Now they are having the two blocks. How does we can do that? By the electrostatic interaction. Now, what will happen? Easily, if you want to go for the genetic delivery system, gene delivery system, it is going to be much easier for you. Plus, polyplexes due to their smaller size may show better transfection. Ready? Now, if transfection is better, you can load your drug. As already I had told, for the effective gene delivery system, it may be a good alternative. If you just functionalize these polyplexes, you can target it to a specific location. And suppose if I loaded it with the SPR, super paramagnetic iron oxide nanoparticles, these may act as a diagnostic agents. Apart from that, for photothermal ablation of the tumor, again we can utilize it. These are the various ways by which just we can utilize the polyplexes as such. Apart from that, again, this is the most fascinating one, I think, nowadays, the dendrimers. Dendrimers are complex macromolecule system. I think everyone is knowing this fact, and they have the critical molecular design for, and which is highly branched structure. These are the cavities where just you can move the drug. On the terminal, just you can move the drug. And at the same time, different drugs can be, drugs and diagnostic agents can be loaded. Right? Water solubility, monodispersibility, presence of hydrophobic and hydrophilic cavities. They are the basic things which is making it more useful for the delivery of different drug materials to a specific location. Suppose if we decorate it with a different type of ligand material, so easily just we can utilize it for the targeting purposes. Currently, the people are using it for diagnostic purposes by loading the drug and different quantum dots at the same time. Everyone is polyamide amines, dendrimers are there, which is especially shown its potential for the oral drug delivery system. Plus, it has been observed that it has enhanced transdermal epithelial permeability. Once you load your drug, the epithelial permeability of your molecule is going to be increased. See, we can utilize it as such. Apart from that, newer generation of dendrimers like the 7th, 8th generation, they have shown their usefulness. Already I had told in the imaging of inner as well as outer vasculatures and the tumor. So we can utilize these things for the diagnostic purposes as such. NIR dyes can be loaded, and in one research it has been reported that 6th generation polyamide amine dendrimer has been utilized for that purpose. Just core was having gadolinium chelate, which is a powerful diagnostic agent. Apart from that, just you can load the NIR dyes. So if you are using two different diagnostic material, you can use it for dual imaging technique or for the multimodal imaging technique. As a imaging modality, just you can utilize it. Number of lipid-based carriers are also there. The nanoemulsions, SLNs, liposomes, the liposomes, neosome, and so on. In case of nanoemulsions, one example just I've included here. Otherwise, number of papers are reporting that just drug and different diagnostic agent can be loaded into that. Here, one scientist, they have reported that use of supermagnetic iron oxide nanoparticle loaded in the nanoemulsion coupled with the erythroma specific monoclonal antibodies, and that just they have utilized it for specifically find out the chances of erythrosclerosis. You can load the espion as such, and you can go for the magnetic resonance. You can go for the magnetic particle imaging. Plus, I think everyone will realize this one. Everyone agree with this one. The nanoemulsions are going to have a better penetrability. So, in terms of penetrability, again, this we can utilize it. Liposomes and neosomes, these are the most explored one. Start from the approval of Doxil, which was the first one. Doxil liposome is the first FDA approved product. So start from that, continuously we are using a number of formulations are already there in the market. We can load the different diagnostic material and you can utilize it for the PET, MRI, SPECT, but whatever the conventional techniques are there, we can increase the effectivity of our contrast aging. Apart from that, one scientist, Richardson, was first utilized the Cancer-99. They have labeled the liposomes for the tumor imaging. In the initial just they have started to do that. Number of papers are there where you can get the surface functionalization of your liposomal preparation for the better targeting purposes. And I think there is no need to tell you the hydrophobic, hydrophilic, both molecules can be loaded. We can just utilize it for the gene delivery system as such. Number of scientists are there who have developed the quantum dots. They have loaded the quantum dots. They have utilized the docetaxel as such. Once you have used a therapeutic molecule and the diagnostic material, Now you can utilize it as a theranostic material. By using these formulations, just we can develop a better therapeutic system, we can develop a better diagnostic system and different theranostic system. Number of papers you may get there. Easily, with the liposome, one issue was the major one which already I had discussed. The RES uptake may be higher. Easily by pegylation you can control that. Once you pegylate it, you can make it move for the more circulatory for the longer time. In the similar manner, just we can utilize the neosomes for loading of different iron oxide nanoparticles, quantum dots, and so on. Similar to the polyplexus, lipoplexus may be there. Here, just we utilize in place of polymer, just we take lipid carrier systems. Yeah. And again, similar to the polymeric system, just we can attach the DNA to it. It's comparatively more easy. Whatever just you can find out in different literatures, you have to mix a lipid suspension to the plasmid DNA and incubate it at the room temperature. And easily the systems can be prepared. Definitely, again, I would say theoretically it's much easier, but in the practical aspect, number of variables may be there which need to be controlled, which need to be optimized first. Here, one scientist, just I have mentioned here, who et al. Have reported ethers cationic lipoplexus nanoparticles, which was facilitated into the diagnostic purposes. So, diagnostic therapeutic agents can be loaded into them. Then, for theranostic system based on the gadolinium nanoparticles and photoporphyrin X loaded neosomal formulation has been reported for serving a dual imaging probe. So these are some common examples where just you can get the basic detail how these formulation, neosomal formulation or the liposomal formulation can be utilized for diagnostic purposes and therapy purposes. And in the initial, I had already told biofunctionalization is comparatively much easier. That's why for the targeting purposes, it can be utilized as such. These are some examples where the people have utilized the different diagnostic material and the drug material as such for these purposes. Number of theranostic applications have been reported in the literature. Then solid lipid nanocarrier system, it may be SLN, it may be nanostructured lipid system. One utility which is very good, that is SLN loaded with gadolinium. Then you have to utilize gadolinium DTP chelate and you can load in the SLN. Similar to the polymeric micelles, it can be utilized for the MRI of colorectal tumor. This formulation is already in the clinical trials and it is expected. Yes, in the future, maybe in the market. At the same time, supermagnetic iron oxide can be loaded and better MRI images you can get. In case of photothermal ablation, this you can utilize that. Suppose you have loaded the spion molecules in the SLN system and you are targeted by functionalization that. Once these spion reaches to the specific location, by radiation, just you can increase the temperature of a specific environment. Because spion are there and due to the resonance, the temperature and everything will get increased, and this increased temperature may lead to the degradation of tumor cells. So for the photothermal ablation and all that, this you can utilize these things too. Sir, I think I have 5 minutes more.
Yes, sir, please conclude.
Yeah. Okay. So in this way, just number of things are there. We can look at quantum dots for this purpose. Number of examples are there where just quantum dots, different drugs has been loaded for the bioimaging and therapy of different type of cancers. Apart from that, number of inorganic carriers, especially the gold nanoparticle, which is just more fascinating, especially when we talk about the diagnostic and therapeutic use in the management of cancer. The gold nanoparticles are going to be used, and especially with the surface functionalization, it can be act as a selective nanopore for specific diagnosis. Even colorimetry, for the colorimetry detection of specific diseases. For the DNA, just you can utilize these things. Similarly, in case of silver nanoparticles, they can be utilized for the different purposes. And in case of silver nanoparticles, it has been clearly observed that apoptosis process get increased. So as the program cells that get increased, obviously your tumor is going to be managed. The number of utilities may be there of nanoparticles. You can prepare different biosensors by utilizing these silver nanoparticles. Then in conjunction with graphene oxide, it may be utilized for the fabrication of different electrochemical sensors for finding out the carcinoembryonic antigens. Apart from that, if any bacterial infection is there, like E. coli, it has been observed that E. coli get adhered at the silver nanoparticle, so it can also be utilized for the detection of E. coli by the electrochemical methodology. Iron oxide is the most cheaper one, and anyone can utilize that. Number of formulation of which is already approved by the FDA, that is ferromoxil, that is coated with siloxane, then ferrocarbotron, that is coated with the carboxydextran. The number of formulations which are already having the iron oxide nanoparticle. As such, iron oxide nanoparticles has been discontinued because of their accumulation in the various body parts. But still in their formulation form plus biofunctionalized form, they are still in use, and they are FDA approved for different purposes. Ferriglucose may be there, ferromoxils may be there. Apart from that, number of things may also be there, like the quantum dots, carbon nanotubes, zinc oxide nanoparticles, graphene-based nanocarriers that can be utilized for these purposes, and we can develop effective nanotheranostics for these purposes. There are number of formulations which are already at the clinical level. Already I have mentioned in the iron oxide particle section. Faradex, SOS, ferromoxtron, Definity, Optison. These are the formulations which are already there and being used for the imaging purposes. Corner dots, Sonaview, Limison. Easily you can find it at the FDA sites. In the initial, I had already told, yes, we are in the process to develop the effective nanotheranostics by utilizing these things. We are in the way, we are developing it, and definitely we are expecting in the near future we can get that. As such, it seems damn easy theoretically, but already I had told number of variables will be there. We need to optimize that. We need to know about the pharmacokinetic behavior of these systems. Until unless we know that, we will not be able to develop an effective therapeutic system, diagnostic system, or theranostic system. So just miles have to go. But again, we hope as we are rigorously working for these things, definitely in the near future, we will have some better results related to the theranostic application of these systems. And more efficient utility of nanocarriers will be there during the development of nanotheranostics. Thank you, everyone. So,
thank you, sir, for this informative session. I request the delegates to interact with the guest speaker. If you have any query, kindly raise your query, please.
Any question?
so one thing I would like to clear, because one student was asking in the previous session how just we can find out if somebody is having the cancer or not. Obviously, cancer as such you can't detect that, or just you can't get to go jump to the conclusion that somebody is having the cancer. But anytime, as the sir was telling, any type of deviation is there in your normal physiology. Suppose a mole is there and pus formation has started there, or it is just having a bleeding pore, or the color of that mole get changed. Somewhere in your body you are having a piercing pain on the regular basis. So that may be an indication to you. Immediately you need to rush to the doctor and just consult it. that is an indication for that. So every time, just be aware, and observation is very important.
Thank you, sir. Thank you very much, sir, for the wonderful presentation on the role of and significance of nano careers for the development of diagnostic in the management of cancer. We would like to give you virtual felicitation to you, sir, for sharing your expertise in this session. Kindly accept our virtual facilitation, sir. Please.
Thank you. Thank you, ma'am. Thank you, sir.
Please give round up. Thank you.
Thank you, ma'am.
Dear delegates who have joined the conference online, feedback link is available. Kindly fill the feedback form, which is available in the chat box. Dear all, scientific session for the delegates who have joined online have to submit. They have submitted their abstract for oral presentation will be held from 2 p.m. onwards. The link for joining the session via MS Team already shared with the delegates through email. I request all the participants to join the session on the time for a smooth conduction. Dear all, that's all for the day. Kindly join us at 2 p.m. for the scientific session in the same seminar hall block C. Thank you.
Students, you also advised to submit the PPT to the Dr. Satish Silpi sir, na, for tomorrow presentation. Dr. Sarath.
Thank you, sir. Thank you, sir.
Achha. Thank you, Dr. Sarath, if you're there.
Thank you, sir. Thank you.